Welcome back to the deep dive. To start things off today, I want you to imagine that you're reading a mystery novel.
Okay.
You're really trying to figure out who the killer is. You've got the book in your hands and you're reading the words, but there's a catch.
There's always a catch.
Right. You're only allowed to read the very last sentence of every single chapter.
Wow. That sounds incredibly frustrating. I mean, you might figure out the general genre of the book, maybe catch a character name or two, but the plot.
Yeah, the plot is completely gone.
The motive, the big twist in the middle, you're completely in the dark.
Exactly. And the reason I'm bringing this up is that according to the stack of research we have today, this is essentially how we've been reading the book of life. You know, the genetic code inside our cells. We've been doing it this way for the last decade.
Yeah, we really have.
We think we're seeing everything, but we're actually just skimming the headlines.
It's a really sobering realization when you dig into the literature. We often talk about single-cell sequencing as this ultimate high-definition camera for biology, but the papers you've pulled for this deep dive, specifically this breakthrough study from Nature Communications published in late 2025, they suggest that our high-def camera has actually been missing about 90% of the action.
Which is terrifying. I
Yeah.
mean, if the biological equivalent of the killer in that mystery novel is hiding in the pages we aren't reading.
Right.
So today we are talking about resolution. We're going to break down a new method called RoEK and ROI that promises to finally let us read the whole book. And then we're going to pivot to the other side of the coin, which is artificial intelligence.
Because once we have all this new high-resolution data, we're definitely going to need AI to actually understand it all. But we have a second set of papers here warning us that our current AI models might be suffering from a pretty dangerous illusion of readiness.
It's basically a perfect storm. Better data meets potentially lying robots.
That's one way to put it, yeah.
But let's start with the biology. I think most of you listening know that single-cell RNA sequencing, scRNA-seq, is the gold standard right now. We use it to see which genes are turned on in individual cells. I always thought this was the absolute truth. So why are you telling me it's lossy?
Well, it is the gold standard for breadth, but absolutely not for depth. To understand why, we have to look at how the machine actually catches the RNA molecules.
Yep.
Inside a cell, you have thousands of RNA messages just floating around. And standard machines like the BD Rhapsody system, which is super common in labs right now, they use microscopic beads to basically fish them out.
Okay, so I'm picturing a tiny little fishing trip inside a test tube.
Precisely. But here is the snag. The hooks on those beads are designed to grab only one very specific part of the RNA molecule. They grab what's called the polyA tail.
The polyA tail.
Right. It's just a string of adenines at the very end of the molecule.
So sticking with my book analogy, it's like grabbing the book by the back cover.
And that is exactly where the sequencing starts. The machine grabs the tail, the 3' end, as we call it, and reads a little bit backwards from there. But RNA molecules can be really long.
Like thousands of bases long, right?
Exactly. So if the mutation that causes the cancer or the splice variant that fundamentally changes how a cell behaves is located at the beginning or even the middle of the molecule.
The machine never sees it.
Never sees it.
It grabs the tail, sequences the end, and just assumes the rest of the molecule is totally normal.
Correct. And we call this the dropout problem or the 3' bias. In standard workflows, we might only be capturing 5 to 20% of the actual transcript information.
That is wild.
The rest is just dark matter. We know it's there, but we just can't see it.
That seems like a massive design flaw. I mean, if I'm looking for a fusion gene where two genes are literally smashed together, that smashing point could be anywhere, right?
Anywhere. And that's why fusion genes are notoriously hard to detect in single-cell data. You might identify what type of cell you're looking at, but you completely miss the disease driver because it's hiding in the unsequenced middle part of the transcript.
So obviously we need a better way to fish. And that brings us to the star of today's show, which is RoCK and ROI. This paper came out in December 2025. First of all, what does the name actually stand for?
It's a great acronym. RoCK stands for Robust Capture of Key Transcripts, and ROI stands for Regions of Interest.
I love a good science acronym, but what is it actually doing that's different? Is it just a better bead?
It's not just a better bead, it's a smarter bead. The researchers realized that standard single-cell workflows basically force you to make a choice.
It's a compromise.
Exactly. You can either get a broad, blurry overview of everything, that's whole transcriptome analysis or WTA, or you can zoom in on a few specific things, but then you lose the big picture of the cell state.
And usually in science, you can't have your cake and eat it too.
But RoCK and ROI is essentially the have your cake and eat it too protocol. They found a way to keep the broad overview while simultaneously deploying a search and retrieve team for specific, hard-to-find molecules.
Okay, let's unpack the mechanics of that. How does the RoCK part actually work? How do they fix the whole grabbing the tail problem?
This is where they get really chemically creative. They take those standard beads I mentioned. Now, usually those beads are covered in two things. The tail grabbers, which are called DT oligos, and another sequence called a TSO, or template switching oligo.
Okay, so the bead has two types of hair, basically.
Right, two types of hair. The researchers found a way to modify the TSO hair using enzymes, specifically T4 DNA polymerase and lambda exonuclease.
Whoa, okay. Without turning this into a massive biochemistry lecture, what do those enzymes actually do to the bead?
Fair enough. Think of the enzymes as a construction crew. They strip back part of the TSO hair and rebuild it into a custom magnet.
Ah, a magnet.
Yeah, so instead of just passively waiting for a generic tail to float by this new capture sequence, actively hunts for a very specific target gene sequence.
So even if the tail is damaged or buried or just too far away, this custom magnet grabs the gene exactly where you want it.
Yes. And the best part for the folks running the labs is that this modification is incredibly stable. They showed these RoCK beads can sit in the fridge for at least 19 months and still work perfectly.
19 months. So a lab could prep a huge batch of, say, leukemia-detecting beads and just pull them out and use them as needed.
Exactly. It's totally scalable.
Okay, so RoCK gets the specific fish in the boat.
Yeah.
But you said earlier that the sequencing machine still likes to read from the end. If RoCK catches the middle of the molecule, but the sequencer still starts at the end, aren't we just stuck in the same place?
That is exactly where the ROI, the regions of interest part, comes in. This is the second half of the hack. During the library preparation phase, when they're getting the RNA ready for the actual sequencer, they add specific primers.
And primers are just little starters for DNA replication.
Right, exactly. But think of these primers as bookmarks. They tell the sequencing machine, hey, don't start at the back cover. I want you to start reading right here on page 42, paragraph 3.
That makes perfect sense. So RoCK grabs the specific book off the shelf, and ROI forces the reader to open it to the exact page where the plot twist is hiding.
That's the synergy. RoCK boosts the capture, and ROI focuses the reading. And when you put them together, the difference isn't just some small percentage point bump. It's a total game changer for the data.
I was looking at the validation data they published, the mixing test. The numbers here are honestly kind of shocking. Walk us through what they did for that.
So they created a controlled experiment to prove this works. They mixed mouse cells that express a green glowing protein, EGFP, with human cells that express a red glowing protein called TD tomato.
Okay, so standard lab stuff.
Right. And they knew for a fact which cells were green and which cells were red.
They had the ground truth.
Exactly. They had the ground truth. Then they ran this mix through a standard unmodified sequencing workflow. The result: the standard method only detected the green gene in 4.8% of the green cells.
Wait, so 95% of the cells that were literally glowing green under a microscope looked empty to the sequencer.
Yes. The sequencer basically said, I see the cell is here, but I don't see any green gene. That is a massive false negative rate.
And when they used the full RoCK and ROI combo,
The detection rate jumped to 99.37%.
from 4.8% to over 99%.
Yeah.
That's not just an improvement, that's a complete revelation.
It fundamentally changes what we can actually see in biology. They also found that the number of reads, which is basically the signal strength for those specific regions, increased by 2,000-fold.
2,000-fold.
It's like trying to listen to a whisper across a crowded football stadium versus having someone scream into a microphone right next to your ear.
Okay, so it works incredibly well in glowing mouse cells. But the paper quickly pivots to real-world applications, specifically in cancer. And this is where I felt the stakes really go up for you as the listener. Tell me about the splice problem they looked at in the colon.
Right, so they looked at a gene called PDGFR-alpha. This gene is really crucial in the gut, but it has two different personalities or isoforms. There's a long version and a short version, depending on how it gets spliced together.
And presumably the standard method just sees PDGFR-alpha and moves on.
Exactly. It sees the tail and says, yep, the gene is here. It cannot tell if it's the long or short version because the difference is completely hidden in the middle of the gene.
Right.
But using RoCK and ROI, they targeted the specific splice junction, the exact seam where the gene was cut.
And what did they find when they did that?
They could map exactly which cells were making which version. They found that fibroblasts at the top of the crypt structures in the colon were making the long version, while the ones at the bottom were making the short one. Yeah, it implies this physical gradient of signaling that likely organizes the whole tissue structure.
That's amazing. It's connecting the physical location of a cell to a very specific molecular decision it's making in real time. But the application that really hit home for me was the leukemia study.
Ah, yes. The BCR-ABL1 fusion. This is the classic genetic car crash that drives chronic myeloid leukemia. Two entirely different chromosomes break and fuse together.
And like we said before, the crash site is the hardest part to actually see.
Right, because the fusion point can vary quite a bit. You have major breakpoints and minor breakpoints, and clinically knowing which one a patient has changes the entire prognosis. So they tested RoCK and ROI on a real patient sample.
Okay.
Now, standard clinical tests like FH, which uses fluorescent probes, they had already analyzed this patient.
What did the standard test say?
They said the fusion gene was present in the lymphoid cells, which is the expected presentation for this specific case.
Okay, so the doctor looks at that and thinks we're dealing with a standard lymphoid leukemia.
But when they ran RoCK and ROI on that same sample, they found the fusion gene in the monocytes as well.
Wait, monocytes are myeloid cells. That's a completely different lineage of blood cells.
Completely different. It means the root of the cancer was much deeper in the developmental tree than the standard tests revealed. The disease was far more widespread in the patient's body than anyone knew.
That is super heavy. It implies that healthy cells might not be healthy at all. They just have low-level drivers that we haven't been able to see until now. It really makes you wonder about the whole definition of remission or disease-free if our tools are only catching 5% of the signal.
That is the haunting implication here. We've been making life and death decisions based on low-resolution data for years. RoCK and ROI finally turns the lights on.
Okay, so we have the wet lab solution. We can finally generate this incredibly deep, high-resolution data. But that creates a completely new problem, doesn't it? Volume.
Oh, absolutely.
We're going to be flooded with petabytes of this complex genetic info. Human doctors cannot read it all.
No, we definitely can't. So inevitably, we have to turn to artificial intelligence to sort it out.
And this is where things get complicated. Because we have a paper here from February 2026 introducing a new AI system called DeepRare.
DeepRare is fascinating because it marks a real shift from chatbots to agents.
What's the main distinction there for the listeners?
So a chatbot is passive. It essentially just predicts the next word in a sentence based on its training data. An agent like DeepRare is active. When you give it a case of a rare genetic disease, it doesn't just guess an answer. It actually goes out and uses tools.
Like what kind of tools?
It searches medical databases, it retrieves similar patient profiles from literature, and most importantly, it has a built-in reflection step.
It checks its own work.
Essentially, yes. It formulates a hypothesis and then pauses to ask itself, does this actually make sense given the retrieved evidence before it gives the final answer? And the performance really shows. Compared to a standard GPT-4o model, which had a recall rate of about 25% for these rare diseases, DeepRare hit nearly 54%.
That's a huge jump. So we're totally good then. We have the super microscope with RoCK and the super doctor with DeepRare. Problem solved.
You would hope so, but we really have to look at this other paper from late 2025. It's titled The Illusion of Readiness in Health AI. And this paper is basically a giant warning sign planted right in the middle of the road.
Yeah, I read this one, and the term visual necessity failure really stuck with me. It sounds super technical, but when you realize what it actually means, it's honestly kind of maddening.
It really is. The researchers wanted to stress test these top-tier AI models, GPT-5, Gemini, those kinds of models. They wanted to know, does the AI actually understand the medicine, or is it just gaming the test?
Right. So how did they test that?
They took actual medical board questions that require an image to answer. So, for example, a question that says, what is the most likely diagnosis based on this X-ray? And it shows a picture of a broken bone.
Standard medical school stuff.
Exactly. But then they removed the image. They just gave the AI the text of the question.
So logically, the AI should just say, I can't answer this. I need to see the X-ray.
That is what a human doctor would do. But the AI models didn't do that. They guessed. And disturbingly, they guessed the correct answer significantly more often than random chance.
Wait, how can it possibly guess the right answer to an X-ray question without seeing the X-ray?
Statistical shortcuts. Maybe the text of the question had a really subtle linguistic cue that correlates with the answer fracture in its massive training data. Or maybe it had simply memorized similar questions from the internet. But here is the part that proves it's an illusion.
What did it do?
When the researchers asked the AI to explain why it chose that answer,
Let me guess. It just made stuff up.
it fully hallucinated a visual analysis. It wrote out detailed sentences like, "I can clearly see a radiolucent line in the distal tibia," when there was absolutely no image file even loaded into the prompt.
This is wild. It's
Really?
not just wrong. It's actively lying to justify being right.
We call it hallucinated reasoning. And they found other fragilities in the models too. Simply shuffling the order of the multiple choice answers, like moving the correct answer from A to C, caused the accuracy to completely collapse in some cases.
So it doesn't actually know the answer is pneumonia. It just knows the answer is usually option B when the question looks like this.
Exactly. This is the illusion of readiness. The model scores incredibly high on the benchmark test, so we think it's ready for the hospital. But under the hood, it's relying on probability tricks, not actual medical understanding.
So how do we reconcile this? On one hand, we have RoCK and ROI giving us this beautiful, deep truth about ourselves. And on the other hand, we have AI that acts like a highly confident medical student who didn't study at all but is just really, really good at multiple choice guessing.
Well, the irony here is that the wet lab solution, RoCK and ROI, might actually be the cure for the AI's problem.
How so?
Think about why AI hallucinates in the first place. It often happens because the model is desperately trying to bridge a gap in the data. If you feed an AI standard single-cell data, that lossy data where 95% of the transcript information is missing, the AI has to guess. It has to infer what's happening in all those dark spots.
Right, it's trying to complete the picture with statistics.
Right. But if you feed it RoCK and ROI data, if you give it absolute ground truth, like we know for a fact this gene is spliced this way and we know for a fact this mutation is present, the AI doesn't need to hallucinate. It has the hard evidence right there.
So the high-resolution wet lab tools are the reality check that keeps the AI honest.
Precisely. We are entering what really looks like a golden era of biological resolution, but it only works if we combine the two correctly. We need the have your cake and eat it too data from RoCK to feed the agentic reasoning of systems like DeepRare.
It feels like we're moving from an era of probabilistic medicine guessing based on statistics to deterministic medicine where we're actually knowing based on detection.
That is the ultimate goal. We want to stop inferring and start knowing.
It leaves me with a thought to wrap up on for everyone listening. We saw in that leukemia patient that the standard clinical test said lymphoid only, but RoCK found the cancer hiding in the monocytes too. That patient's entire treatment plan would have been based on completely incomplete information.
And that's just one patient in one study.
Right. So if an AI can guess a diagnosis without seeing the X-ray, and a standard sequencer can miss 95% of a gene, how many people are walking around right now labeled healthy or in remission simply because our resolution wasn't high enough until today?
That is the question that keeps me up at night. But hopefully with tools like this, we won't be in the dark much longer.
Biology is in the details, everyone. And it looks like we finally got a better microscope. Thanks for listening to The Deep Dive.
See you next time.